Hello, my name is Brendan Nicholas, and this is my co-host, Baby Lily, and this is Science News.
Audio Jungle.
Cloning produces human embryonic stem cells. Fine-tuning of technique used in other animals could enable personalized medicine. For the first time, scientists have created human embryonic stem cells by transferring the nucleus of a mature cell into an egg. Researchers suggest the cloning technique could nudge the dream of personalized medicine closer to reality. Yes, it's a huge landmark achievement. Creating human embryonic stem cells by nuclear transfer in humans is the next major technological advance since Dolly. The famous sheep Dolly was the first mammal cloned by nuclear transfer technique, in injecting the nucleus of a cell from one adult sheep into an egg of another. Since the animal's birth in 1996, scientists around the world have tried to duplicate this technique in human cells. Unlike adult cells, which already have followed a path to become, say, heart cells, neurons, or skin cells, embryonic stem cells are uniquely poised to become any cell in the body. And if scientists can make these stem cells from a patient's own tissues, once untreatable conditions could perhaps be cured by replacing damaged cells with healthy ones. But creating embryonic stem cells in humans has proven tricky, said Catherine Platt, a stem cell biologist at the University of California, Los Angeles. No one knew why the technique worked in other mammals, but not humans. Researchers had to figure out the best way to ease out an egg's DNA, slip in a new nucleus, and then cue the egg to divide and grow. In 2011, scientists came close, but the egg stalled out after 3 divisions, producing just 8 cells. In 2007, a new way to make stem cells dazzled scientists in the field. By dosing human cells with a small cocktail of molecules, researchers pushed a reset button that turned adult cells back into embryonic-like ones, called induced pluripotent stem cells, or iPS cells. For the last 67 years, virtually all of us has ended our nuclear transfer efforts and switched over to iPS cells. But a team led by Shaukat Mitalapov of the Oregon National Primate Research Center in Beaverton kept plugging away at nuclear transfer, first using the rhesus macaques and then using human cells. One key change was adding caffeine to the eggs before DNA transfer. Caffeine acts like a set of chemical reins, holding back the eggs' development until researchers inject a new nucleus. The new protocol also features other tweaks, such as examining the eggs under polarized instead of ultraviolet light, which can damage the egg. Using the new method, researchers made embryonic stem cells from an egg and the nucleus of a young boy's skin cell. The new cells can grow and divide to form a mass of embryonic stem cells, just like those derived from fertilized embryos. And when researchers ground the cells up and compared their genetic bits to those in embryonic stem cells, they didn't see much of a difference. Virtually all of the new cells' genes were reset to their embryonic states. What's more, the approach boasts dramatically improved efficiency. Instead of burning through thousands of eggs to make a single embryonic stem cell line, our group could use just 2 eggs. Well, the new cells may have advantages over iPS cells, treating some genetic flaws that lurk in mitochondria, little cellular power plants that carry their own DNA. By putting the nucleus of a patient's skin cell into a fresh egg with healthy mitochondria, scientists could potentially make a customized therapy that erases the defects. My researchers ought to compare the new embryonic stem cells with iPS cells. A side-by-side look might provide clues to how resetting adult cells actually works. If they can figure out why Mitalapov's nuclear transfer method is so successful, researchers might be able to improve the technique to make iPS cells and avoid having to retrieve from volunteers. Embryonic stem cells using this method have the potential to treat spinal cord injuries and diseases such as diabetes or Parkinson's. I am very confident that such cells will be used for therapies in humans in the near future. Researchers use a modified technique to create human embryonic stem cells using adult cell nuclei and egg cells. The scientists were then able to turn the immature cells into a variety of tissue types, like, say, heart cells. My name is Brendan Nicholas, and this has been Science News. Hello, my name is Brendan Nicholas, and this is Science News. And this is my They should just name you. Oh yeah, it's a huge landmark achievement. Okay, it flies away. So, oh yeah, it's a huge landmark achievement. Creating embryo. Okay. Yeah, it's a huge landmark achievement, guys. I can't do it. Okay. Okay. Oh yeah, it's a huge landmark achievement. Creating embryonic stem cells by nuclear transfer. Embryonic stem cells using the method have the potential to treat spinal cord injuries and disease.